So I prepared this talk also very much in the spirit of wanting to be optimistic. I think it's the scary stuff that gets the news, but what I want to tell you is about some of the ways in which AI applied to our research is helping us make progress faster, more efficiently, and to ultimately get more insights in our particular biological question. So the first thing is to kind of reiterate some of the points that Franca and also Ireleni have also made about the scale of the challenge in biology and what we might say one of our goals in biological research is. And one way of thinking about it, if we wanted to be very ambitious, is to say that to understand the health of a human, we would like to understand the behavior of every atom from which their body is composed. So that's a very, very big data problem. And I'm not going to be able to tell you about all the atoms in my body today. But what I'm going to do is try and explain some of the ways in which we're starting to unpick this big problem and how helpful AI applications have been for us in that process. So just to remind you again about the hierarchies of biology, sticking with biology rather than subatomic matter for this evening, we start with our atoms that go together, for example, to make up the DNA that composes our genes. Our genes are going to inform our cells in order to make the proteins that Ireleni has already explained to you. And this is at the nanometer scale, so that's a billionth of a meter. And when we put all of those proteins together in the context of a cell, we get something that looks recognizably like something you might see in a biology textbook at school. When we put all those cells together, we start to build an organ. And when we've got enough organs together, we will ultimately have the human body. And medical science could be said to be aimed at understanding the way healthy humans work and what goes wrong in disease. And so if we frame our biological questions in that way, we can start to understand that we've got a lot of data. And because biology is noisy, we've got a lot of noisy data, and computers have always been very good at helping us to dissect that and to dissect that with increased efficiency. So putting it a different way, I don't think these cars are even on the road anymore, but if this is the engine that we're overall interested in getting to run, one way in which you might like to understand how it works is to take it apart. And that's one of the ways in which biology can be said to be working is, maybe we might consider that understanding a whole human body is rather a complicated thing, but if we take all the parts, keep track of them, we can start to understand individually how those parts work, and then we start to put them back together again. So this is the machinery that I'm going to be referring to, and in the context of my research, the machinery is the machinery that keeps our cells alive. So this is kind of the scale at which we're working. And the type of cell that we're particularly interested in is the brain cell. And I'm very now conscious, given Ireleni's beautiful talk, that I'm going to be talking to you about how neural networks help us understand our neural networks in a way. But ultimately what we would like to know is what is it that allows our brains to form accurately and healthily, and what goes wrong. And there are lots of, as we unfortunately know, lots of examples where the brain function goes wrong. And if we understand what goes wrong, can we then start to think about interventions that can rescue some of those errors? And I'm going to give you a little bit more brain biology before we go into the research that my group is interested in. So this is now one of the building blocks of the brain. So these is one of the neurons from which our brain is composed. And remember, Ireleni told us that there are billions of these inside a human brain. So we narrow, for our purposes, we narrow our focus in on individual versions of this kind of neuron. And in particular, we're very interested in the organization inside, so the machinery inside this neuron that makes it function. And in particular, my research is interested in the structural elements inside these cells that help them both maintain their special structure, which conveys the information around the brain, and for example, helps them move during brain development, because there's a lot of neuronal migration as the human brain develops. So one very clear and very tragic example of that is a set of diseases which are called Lissencephaly. And you'll be probably, I think, quite familiar with images of a healthy adult human brain in cross-section. And one of the things that we're very familiar with seeing is the folding on the surface of the brain, which facilitates accommodation of the billions of neurons from which our brains are built, and also the layering within the outer edge of the brain, which also facilitates compartmentalization and connectivity between particular populations of neurons. So this is how evolution has allowed our bodies to develop in order to form this particular structure. And in patients with Lissencephaly, various aspects of this process has gone wrong, and it's gone wrong early in their development, so that when these children are born, they're missing very, very many important aspects of a well-formed human brain, and so they experience a lot of suffering, and they don't live for very long because of this profound effect. Essentially, what research by a number of groups has shown is that what goes wrong is that the neurons that form ultimately this healthy brain start in the middle and they move outwards. And what happens during the development of a Lissencephaly patient is that basically the neurons get lost on the way. There's, as you can imagine, it's like the absolute worst of a London rush hour, all these neurons trying to get to where they need to get, where everyone else is, all the other neurons are trying to get to a slightly different place. But there are very sophisticated guidance mechanisms inside the neurons that don't work in the Lissencephaly patients. And one of the components of that guidance system relates to the underlying scaffolding that supports the shape and movement of the neurons. And that's what we're trying to understand. So there are two ways in which I want to tell you a little bit about how AI is helping us start to get to the bottom of what's going on. The first one is a data management question, basically, because if we want to understand ultimately where all the atoms are in the scaffold, inside the neurons, inside the brain, then we would like to understand where everything is. And in order to be able to do that, we need a lot of data in order to extract that kind of detailed information. So that's one part of the research I'm going to tell you about. And the other one is to try and understand that scaffolding inside a cell. And in that context, what's really challenging about those experiments is that, as well as the scaffolding that we're interested in, and which we know is disrupted in disease, there's everything else in the neuron as well. And so we need help from the computational analysis in order to extract the things that we're interested in, to try and integrate our understanding of what could go wrong in the context of that kind of neuronal and developmental disease. So the particular method that my group uses involves using a very powerful microscope, and it was a type of microscopy that was recognized in the 2017 Nobel Prize for Chemistry. Three pioneers in the use of an electron microscope data were recognized, and there was, of course, a great deal of work that led up to the award of that prize, and also the application of that methodology to many, many different kinds of biological problems. If some of you stay around for the tour, you will be able to see one of our most powerful microscopes. Here's a human for scale. It's a very big microscope, but what's great about it is that it's very powerful, and it does, in principle, allow us to see the individual atoms from which the molecules that we're interested in, or the cells that we're interested in, are built. I'm going to refer to it as cryo-EM. That's because the sample is frozen. That helps us in some ways in our experiments. As I said, the microscope is very powerful, but because of the nature of biological samples, the data are also very noisy. So again, the use of computers in our experiments is very helpful to us. So just to remind you again where we're interested, we're inside a neuron, and we're interested in the scaffolding inside the neuron that supports its shape and also its movement during development. And I'm going to tell you a little bit about what those scaffolds are built of. You won't be tested on this afterwards, but these building blocks are they are made of a protein called alpha-beta tubulin, and alpha and beta are two different proteins, but they always come together. We know quite a lot about the individual structures of alpha and beta tubulin, but we would like to understand how they work together, and this is where our electron microscope experiments come in. So if you wanted to build a microtubule, what you would need is lots and lots of these subunits, and they are they have evolved to build microtubules in a coordinated way. So they connect to each other in a head-to-tail manner, so sort of a top-to-bottom way, and they make connections side by side. So if you have enough of them, what you get is a hollow cylinder of alpha-beta tubulin subunits, and this is the scaffolding element called a microtubule, and neurons are absolutely jam-packed full of microtubules, and they do very many very important things to the neuron to support neuronal function. So what we also know is that microtubules are very dynamic, which is to say that they can put themselves together by forming these connectivities, but they also self-destruct. And for this reason, neurons also have evolved very many regulatory proteins that help stabilize microtubules under particular conditions. So that brings me to the particular component of the neuron that we know, when mutated, causes Lissencephaly. So this particular protein is called double cortin. And just to emphasize the scales again, that Lissencephaly can sometimes be caused by a single tweak to a single base in the genome of the patient with Lissencephaly. That tweak causes a change in the double cortin protein, which causes a change in the behavior of the neurons in some way that we don't understand. And when that's amplified into the billions of neurons inside that developing brain, it stops working. We don't know why. So this is what we would like to understand. What we do know is that this protein called double cortin can act to stabilize microtubules, and there's quite a bit of evidence that the stabilization property of double cortin in microtubules is very important for brain function. And what we also know is that in the absence of double cortin, it appears that the microtubules inside neurons become fragile. And so our idea is that that's the microtubule fragility that's really causing ultimately problems in neuronal migration and ultimately in brain development. So this is our idea, and this is what we're trying to get more data about. So just to introduce to you what we know so far, we know that double cortin binds to alpha-beta tubulin in this way, and it sort of acts like a staple in between the individual subunits of a microtubule. So if you have enough of it, then you have a very stable microtubule, and it seems that might be a good thing for brain development. So we've been working on this problem for a while, and in the olden days, when there wasn't very much data around, there wasn't so much that we could really say about the way that double cortin was working, but increasingly we have more and more data, and we get more and more information about how it works. So this is a picture that comes out of an electron microscope of microtubules. In my opinion, microtubules are one of the most photogenic things in biology. But I probably would say that. But just to give you an idea of how powerful that microscope is, this is an individual microtubule. If you squint, you can kind of see the little indentations in the wall of the microtubule, and those correspond to the individual subunits from which it's built. But this is the diameter of this tube, this cylinder, is 25 nanometers, so it's very, very, very small. But this is what we would like to try and understand. So when I first started working on this problem, a long time ago. This was the view that we had of double cortin. So this, you can roughly see that this is the wall of a microtubule, and this blue thing is roughly where we thought it sat, but we didn't know very much else about it. So there's not very much information here. It's all kind of smooth and nice, but we don't see any of the details, and particularly we don't see anything about the atoms. But as we gradually got better, we learnt more and more about how double cortin was interacting with the walls of the microtubule, and increasingly as we get more data, we can see even more precisely how the walls of the microtubule are put together and how double cortin is binding to them and holding them together. And what I will say, because Irelenia explained how AlphaFold works so nicely, this is a structural biology problem, but if you give AlphaFold at the moment, you give information about double cortin and tubulin by itself, it doesn't do a very good job of putting them together to show this structure. So there are, as Irelenia explained, there are very many more complicated structural biology problems where you still need to do the experiment. In particular, because microtubules are dynamic, and we know that there are lots of different kinds of inputs that can influence their structure. I think it's still going to be useful for us to do these experiments for a little bit longer, although who knows what AlphaFold will be up to in the next few months or weeks or years. For now, this information came from our experiments coming out of our electron microscopes. And we want to understand that the nature of those interactions with as much detail as we can. And to illustrate that, I go back now to this kind of blurry overview. This is the view of a microtubule, wall, but you can see it's all nice and shiny. But it lacks detailed information. So we see the shape, but we don't see the detail. But as you include more and more information, you get to see more and more detail until gradually you see ultimately where all the atoms are from which that microtubule is built. And the reason why that's important is that those mutations that are affecting Lissencephaly patients and causing their disease are in some cases happening at the level of individual atoms. There's very, very small changes that can sometimes precipitate all of those catastrophic events in brain development. And also we know that the very best drugs bind and are precise at the level of the atoms from which they're composed. And so we would really like to understand where all the atoms are, and therefore the more detail we can get from our structures, the better. So this is now an example of the type of experiment that we use AI for in our molecular experiments. And what I'm showing you here is, again, it's a micrograph of microtubules, which you should be getting the hang of by now. But what the coloured lines are is a neural network program that has taken the thousands of images we collected in this experiment and identified the microtubules that we are interested in, and will take them into our computational programs in order to allow us to determine the structures much more efficiently and with a massive amount of data to allow us to get much more information from our experiments. And when I was a PhD student, we used to do this by hand. So this is which was fine when I only had 30 micrographs. It's absolutely not fine for my team now when there's 10,000 micrographs. My team are much better thinking about how to design the next experiment better, because they do that extremely well, and that they delegate this very boring and very time-consuming job to the AI network that allows them to extract the best and the most highest quality data in order to feed into our structural experiments. So that's one example of how we're using AI. And this is just to illustrate. So this is the structure of two of the microtubule binding components, the folds that Irelenia was talking about in double cortin that interact with microtubules. And on each of these structures, I've mapped the points mutations, which is to say the small changes that cause Lissencephaly in different Lissencephaly patients. What we hoped when we got this structure was that would allow us to understand which patients might be sicker compared to others. There might be useful things that we can do with that. And at the moment, I'm afraid to tell you that we don't currently understand the link between this particular change being particularly bad and this particular change being not so bad. But this is one of the ways in which we want to continue working on the structures of these complexes in order to understand better how disruptions in these structures can have such a profound effect, first on the microtubules and then on the neurons and the brains. But speaking of the context in which those complexes are working, we're also trying to understand what's going on with the microtubules inside the cells. All the experiments I told you about so far were using microtubules that were made in a test tube just from basically alpha-beta tubulin and double cortin. But now really what we want to understand is what's going on with the microtubules inside neurons. So what we do is we take individual neurons and grow them on the surface of our I guess it's a sample holder that can go inside the electron microscope, and there are some particular requirements for that sample holder. And so this is what an individual neuron would look like, and this is what the tail end of a neuron looks like inside an electron microscope. So the blue line sort of outlines the very end of it. And there's a particular reason why we're interested in this part of the neuron, because we know that double cortin is expressed in this part. And then we look inside even further, and now we start to look at, well, this is probably a microtubule, but we don't know. But there's all this other stuff as well, because I said, inside the neuron is everything the neuron needs, but we don't know much about what all of that is. So the other challenge that we set ourselves, and then we asked an AI program to do it for us, is to work out what everything is. So this is an extract of some of the work that we did, in which, as Irelena explained, the process is we first have to explain to the AI program what our question is, and to do that we train it and explain that some things, for example, are microtubules and some things are not. And it churns away on that so-called training data. And then we give it our actual very noisy data, and it begins to work out what everything is. So now we can go in much more efficiently, collect very many volumes from inside different parts of a neuron, and then start to ask ourselves what's going on with the microtubules, what's going on with other components of the neurons, and integrate how all of those things work together, which ultimately is driving what the neuron is up to. So when you start looking inside cells, it really is a bit mind-blowing, because there's just all this stuff, and apparently the cell is organized it, and it's just there doing what the cell needed it to do. But we really don't understand how all of that works. So we start to take a little bit of extract the things that we understand a bit better and start to try and work out what's going on. So what we're now looking at is a very thin slice through the middle of a part of a neuron. And just for ease, we've color-coded the microtubules in pink. And some of this is a mitochondria, which is the energy-generating organelle of the cell. And then we've got some membrane components. But if we focus in on the microtubules, one of the things that we were fascinated to observe, we weren't the first people to see it, but it's really amazing when you see it. I said to you that the microtubules are hollow cylinders, but in the cell, there's stuff inside. And there's all kinds of different shapes and sizes, distributions, and we really don't know so much about what these things actually are. There's quite a big volume overall. I told you that neurons are full of microtubules, and therefore there's quite a bit of space taken up on the inside of the microtubule. In our experiments in the test tube, we typically tend to see nothing, but in cells there's a bunch of stuff, and we genuinely don't know what most of these things are. So that's certainly food for thought. But what we also observed was, so our experiment was to take neurons that did have double cortin and neurons that don't have double cortin, and compare what's going on with the microtubules. Because remember, double cortin binds to microtubules, so we hope to see something. In both cases, we see that microtubules have the same kind of overall organization. They're built from these 13 lines of tubulin subunits that polymerize longitudinally. But what we did see is that in neurons that don't have double cortin, we see many more of these ruptures in the overall structure of the microtubules inside the neurons. And that's quite a sort of consistent thing, both in terms of the number of ruptures and the sizes of the ruptures. So that's the starting point for our idea that double cortin might be involved in supporting the overall integrity of microtubules, and in the absence of double cortin, maybe those microtubules are fragile, which is particularly important in a migrating neuron, and maybe that's the reason why ultimately in the context of a Lissencephaly patient there's such chaos occurs during brain development. But that's a big idea, and now of course we have to do the experiments to prove it. So we're of course extremely happy to have AI at our disposal to take out the boring bits so that we can think hard about how to do the best kinds of experiments to try and get to the bottom of this very interesting biomedical problem. So this is what I hope I've explained to you why we're excited about AI as applied to our research. And I do think if you can scale up those sorts of experiments in a reasonable and sensible way, then it really opens lots of doors in terms of our understanding of what biomedical questions we can really answer. Obviously, none of this would be possible without the fantastic people who are actually doing the work. I've talked about it, these people are doing it. This is the current team, and I want to particularly emphasize that the molecular work that I showed you was done by Shimon, and the neuron work that was done by Joe. None of this would be possible without a fantastic facility staff, at least one of whom you'll meet if you go on the tour. And the computing, you can imagine, is very important, and we already talked about that a little bit. And of course, our collaborators are very important too. I have a movie that I will just play, but I'm very happy to take questions. I think I've distracted people with the movie at this point.
Thank you very much, Carolyn. Do people have questions for Carolyn? I'm just going to check to see if anyone has emailed one. When you're training to recognize microtubules, how much training data do you need to have?
Yes. So that we did, when we went in, we didn't know what we would need. So basically we added more and more data until we got out what we were expecting. So it was very, very closely supervised. So this is an example of where you kind of know the answer that you want, and you have to work with the machine until it gives you what you're actually expecting. Yeah. Yeah. Yes.
Are you looking at whether like the ruptures of microtubules cause stuff to fall out?
So far in our data analysis, we didn't see any connection between the presence of a hole and the presence of the stuff. So certainly many people are working on this very interesting question, and there's some components that are known to sit inside a microtubule, and those are attached to the wall. So the presence or absence of a hole wouldn't make a difference because they're stuck on. So we don't have any evidence that that's the case, but of course, as we want to be open-minded, so as we get more data, we will pay attention to that. And again, that will be one of the things that we can sort of delegate to the machine annotation and just monitor whether or not there genuinely is a difference or not. Yeah.
Any more questions for Carolyn?
There may be some people who know this already, but there's a corner of the internet that says that our consciousness runs through microtubules. I have to say we haven't seen it, but I also don't know what it looks like. So we also remember to think about training our data on some unknown elements inside microtubules. There are many.
Is it not possible to at least know if the stuff inside is nucleic acids or amino acids?
Yes, it is. So there's work by other groups that have, amongst other things, they've seen much more similar stuff. inside microtubules, and they have been able to get more information about the types of things that it might be built from. I have to say, that was not our experience. We saw a very big range of sizes and positions. So I can guess that might be one of the things that very much depends on where the neurons came from to start with, how they were grown, etc. But we genuinely don't understand that. But yeah, there are some experiments that have been conducted in order to try and establish what at least a subset of those things might be. Looks like proteins.
Can I just ask then?
Yeah.
Because, so I don't know what your time resolution is possible, but the mere fact that it's a tube implies things flow through it.
Yes.
Is there evidence for anything moving inside as opposed to just stuck?
Yes, there is very good evidence. So one of the things that I didn't talk about is that there are ways to chemically modify alpha and beta tubulin. And there are all kinds of enzymes that do those chemical modifications. And some of those chemical modifications are on the inside. So there are a known subset of enzymes that must migrate inside in order to access the sites where they chemically modify. So at least a subset of the blobs that we see are probably those enzymes, but by no means the only thing.
Thank you. So a lot going on.
Yes.
If anyone does have any more questions, perhaps we could have some after the
sure.
discussion. Yes. And also, again, if anyone's at home and they want to email in, it's k.thompson@bbk.ac.uk. And can we just thank Professor Moore?